To finish our discussion of the catabolic pathways, we need to discuss how amino acids are oxidized in the body. In this particular chapter, we'll look at what happens to proteins and amino acids as they are digested and degraded for the purpose of producing energy. But we need to remember as we're going through that amino acids are actually our last energy source we will use. Proteins are only broken down when there really is no other option for some sort of metabolite we can break down to produce energy. There are times when we will have leftover amino acids. So as we have proteins in our body, they typically will have normal, what we call protein turnover, where the protein will live its lifespan, which is determined by a particular protein, and then that protein will be broken down so that new proteins can be produced. Once we break down proteins, we will try to reuse amino acids so that we're not being wasteful, but there are often times when amino acids are left over from that. In that circumstance, we will break down those amino acids, potentially making some sort of energy or fuel from those. Another way we will sometimes see amino acid degradation is when we have dietary protein that exceeds the needs that our body has. If we eat more protein than we need, then we will begin to break those proteins down to produce energy when needed. We'll see this very often when people have an extremely high protein diet. We'll see an increase in the amount of amino acids broken down. There are times in our body when our proteins can be broken down specifically for energy purpose, but this is when carbohydrates as well as fats are going to be very scarce. Some examples of circumstances that could lead to protein breakdown in the body would be starvation, certain types of diabetes, and any similar conditions. What we need to think about in this chapter as we go through is not just how we break down amino acids for energy, but what we're really looking at is how our body handles amino groups. Okay, so whether we're thinking about intracellular protein that's being broken down because our body has no other energy source, or leftover amino acids, or if we're looking at dietary protein, we need to think about two things occur when we break down amino acids. We will get carbon skeletons from the amino acids that can enter into the citric acid cycle, ultimately producing energy and or going into gluconeogenesis to produce a glucose for organs like the brain that have no other option. The other important thing that we have to consider is what do we do with this amine group? Now, if we can at all use the amine group, we are going to use it. You may have learned in other courses about nitrogen fixation and studied some things about the nitrogen cycle. Humans can't fix nitrogen, so we try not to excrete any more nitrogen than we need. We try to use anything that we can. The way that we do excrete excess nitrogen in our body is converting the amine group into a molecule called carbamoyl phosphate that can enter into the urea cycle, and we can produce urea for excretion. The last thing we'll discuss in this chapter is this aspartate arginosuccinate shunt that shows you how the citric acid cycle and the urea cycle are linked. So let's first take a look at dietary proteins. As dietary proteins are ingested, the breakdown of protein begins in the stomach through the work of an enzyme called pepsin. Pepsin begins to cut large proteins into peptides. As the smaller peptides continue into the small intestine, we release other enzymes, trypsin, chymotrypsin, aminopeptidase, carboxypeptidase, various enzymes that will further break the proteins down into smaller peptides. Once the proteins are broken down into small peptides and amino acids, those amino acids can be absorbed through the small intestine and go into the bloodstream. Once we get those ingested proteins into the liver, that amino acid is then going to be converted into the carbon skeleton called the alpha keto acid. The amine group from the amino acid we've ingested will combine with alpha ketoglutarate, producing glutamate. Glutamate is a very common carrier that we'll see. Sometimes that glutamate can release the amine so that it can be excreted through the urea cycle. Sometimes that glutamate will combine with another amine group, forming glutamine. This particular picture also shows you two other amino acids that we're going to pay special attention to in this chapter. We will see the link. Very often alanine is the shuttle amino acid we use to get waste nitrogen from our muscles. And then glutamine is going to be used to carry from muscle as well as other tissues. In this chapter, we'll talk a lot about transferring amine groups from one molecule to another, so we need to look at the main enzyme responsible for this particular type of reaction. They are called aminotransferases. The aminotransferase is always going to be named by the amino acid that is donating the amine group. Very often, this aminotransferase is going to use alpha-ketoglutarate as an acceptor for an amine group, which will form glutamate. Glutamate is our primary storage we use in our body for amines. The very common aminotransferases that we see in all these reactions all use pyridoxal phosphate as a cofactor. So I think it's worth noting how the structure of this pyridoxal phosphate. Pyridoxal phosphate is shown here in this picture. It is derived from vitamin B6. This particular PLP molecule can then be aminated and we get the pyridoxamine phosphate. So the PLP will serve as an intermediate in the enzymatic reaction with any of the aminotransferases. The aminotransferase will transfer the amine group from the donor amino acid to the PLP. PLP will hold the amine group until it is transferred to the ultimate acceptor. Now let's look specifically at how ammonia is collected in glutamate. So we're looking specifically at the glutamate dehydrogenase reaction. Glutamate dehydrogenase is an oxidative deamination reaction that only occurs within the mitochondrial matrix of cells. This reaction requires an electron acceptor in the form of NAD or NADP. Glutamate, shown here, with its amine group, will transfer its amine group out into the cell, releasing alpha-ketoglutarate. Alpha-ketoglutarate is the alpha-keto acid of glutamate. This glutamate can travel around the body holding this amine group until that amine group is needed for something else. When that amine group is needed for something else, then the amine will be released. Glutamate will form alpha-ketoglutarate. Glutamine is used to safely transport ammonia through our bloodstream very often. To produce the glutamine, we take a glutamate that has already received one amino group from a reaction such as alpha-ketoglutarate conversion to glutamate, use glutamine synthetase to form glutamine, where we're ultimately adding a second amine group to our glutamate. This reaction is going to occur mainly in the liver, but it can occur in other areas where we will then take that amine that we've added, so we take the glutamine, convert back to glutamate. So we're releasing that excess amine group, still have the glutamate. So I think this is a point where students will often get a little tricked up with this. You have to think about glutamate and glutamine both as storers and transporters. So glutamate is your more typical molecule that is going to just naturally carry around the amine groups. Whenever we see glutamine, that typically means we have excess amounts of ammonia, and that excess ammonia has been added to glutamate, forming glutamine, so the glutamine can then travel through the blood till we get to the liver. Then the glutamine will excrete the ammonia into the urea cycle. Our last look at how we can use specific amino acids to our advantage is looking at what goes on in our very active skeletal muscles. So in our skeletal muscles, we will very often convert the majority of our glucose into pyruvate. That pyruvate can then be used to enter citric acid cycle so that we can produce energy, things of that nature. When our muscles are vigorously working, the pyruvate will begin to slowly convert into lactic acid. When we get this pyruvate buildup, some of the pyruvate will be converted into alanine. To produce alanine, a glutamate that came from muscle breakdown, muscle protein breakdown, will donate an amine group to pyruvate, forming alanine. The alanine can travel in the blood to the liver. In the liver, the alanine is converted back to pyruvate, releasing the amine group into the urea cycle. This is a great example of what we see with people that work out a lot. If you do a lot of weightlifting, you'll hear a lot of talk about tearing down and breaking down the proteins, the muscles, so that we can make the muscles larger, more efficient, things of that nature. If someone is actively working out, there are proteins that will be degraded. Those degraded proteins are releasing amino acids and therefore releasing amines that we need to process. The muscle can't get rid of those amine groups, so the alanine is going to carry that amine to the liver where the amine can enter the urea cycle. As far as the nitrogen use in various organisms, we are going to excrete in the form of urea. So once we form urea inside of the liver, that urea is going to enter the bloodstream, and then we will excrete it in our urine. Birds, reptiles, they, instead of using just urea, they use uric acid. Aquatic vertebrates, fish, they're able to release the ammonia directly. So now that we understand that we use alanine, glutamine, and glutamate as our carriers for amino acids throughout our body, let's look at the actual urea cycle and how we can get rid of excess amine. Okay. So one thing we can look at is if we have this alanine that's coming from our muscle, the alanine can donate an amine group to alpha-ketoglutarate, forming glutamate. Glutamate can go into the matrix within the liver. We can also see glutamine traveling from other tissues, bringing the amines back into the liver. The glutamine will donate an amine group. We get glutamate again, releasing free amines. If we have, we'll come back to this in just a second. So the initial thing we need to understand is, where did this amine come from? Okay. Once we have the amine inside of the mitochondrial matrix of the liver cells, that amine can combine with a bicarbonate using 2 ATP molecules and form a molecule called carbamoyl phosphate. This is a molecule you need to recognize the structure of. Oxaloacetate shows us here, it's kind of another preliminary step within the urea cycle. Oxaloacetate can grab excess amine groups and form aspartate. Now this is going to be important because the aspartate is going to be part of the urea cycle. So you can kind of think of what's going on this slide as, I call them preliminary steps, things that must happen for our urea cycle to get going. Okay, so the 2 steps are, we need to get the amine from other areas in the body, so we use alanine, glutamine, glutamate to do so. And then we also need to prime the urea cycle by converting some of our oxaloacetate from the citric acid cycle, gluconeogenesis, other areas, and convert that into aspartate. So just a little bit closer look at the production of carbamoyl phosphate. Initially, an ATP molecule is going to react with a bicarbonate, forming an intermediate with a phosphoric acid anhydride, very high energy bond. That high energy bond is then going to break. The breakage of the high energy bond gives us the ability to combine the ammonia with our molecule, forming carbamate. We then use our second ATP to phosphorylate carbamate, forming carbamoyl phosphate. Carbamoyl phosphate is the initial molecule that's going to enter into the urea cycle. Here is our total urea cycle. So what I've done for you on this slide is kind of listed by the steps that are written here, 1, 2, 3, and 4, what's going on. Okay? So in step 1, we're going to form citrulline from our carbamoyl phosphate and ornithine. This is very similar to the citric acid cycle. We're going to use an ornithine at the beginning, and then when we finish, that's what we're going to make. Okay? So step 1, With the enzyme ornithine transcarbamylase, we combine carbamoyl phosphate with ornithine, forming citrulline. We then come out of the matrix. Then the citrulline is going to form, ultimately, arginosuccinate. This particular step is going to require an aspartate to come in. And you'll notice we have an amine group from the aspartate, which is now part of our arginosuccinate molecule. The other amine group that we see here in blue, that was the original amine that came from the carbamoyl phosphate. In step 3, arginosuccinase is going to convert arginosuccinate to arginine, releasing fumarate. Now that fumarate should look familiar to you. Remember that fumarate can now enter into the citric acid cycle. Arginine now has multiple amine groups. We release 2 of the amine groups in the form of urea in step 4, and arginine is converted into ornithine, forming ultimately what we started with in the reaction. So the overall goal of what's going on here is we are gathering amine groups. We gathered an amine group here. Remember that we pulled from either alanine, carrying it from the muscle, glutamine, carrying it from other places in the body, and we pulled this amine group from aspartate. We pulled both of those amine groups into molecules, allowing us to release both of those amines into a waste product called urea. It should be quite easy to see just from the molecules involved that the urea cycle and the citric acid cycle are heavily related to each other. So shown here in this picture, you see this is the urea cycle, and this is the citric acid cycle occurring in the mitochondrial matrix. There are often times when the oxaloacetate from the citric acid cycle will be converted to aspartate. The aspartate can then be used to enter into the urea cycle, donating an amine group. When we release the amine group to the urea cycle, we again form oxaloacetate. Oxaloacetate can be converted into malate. Fumarate, also released from the urea cycle, can be converted back into malate. Malate can enter back into the mitochondrial matrix, jumping back into the citric acid cycle. What you should be able to see from this is by using this malate aspartate shuttle and this aspartate arginosuccinate shunt, we are able to constantly fuel these molecules needed for the urea cycle. By using the oxaloacetate from the citric acid cycle, if we didn't have a way of putting things back, it would disable what the citric acid cycle was trying to do. So we can pull just the aspartate off, but at the same time put something back so the citric acid cycle can continue. The urea cycle is heavily regulated by the amount of amines that are available. Carbamoyl phosphate synthetase is activated by the presence of acetylglutamate. High amounts of glutamate tell us that we have a lot of glutamate, we have a lot of amine. High amounts of acetyl-CoA are also going to be involved in activating the urea cycle. If we have high amounts of acetyl-CoA, that means our body is actively metabolizing. These urea cycle enzymes are also regulated by expression. Anyone that eats a protein diet, a diet that's extremely high in proteins, they are actually going to make more enzymes to create the urea cycle and make it continue, because the urea cycle is really most important for breaking down proteins. Also, people that are constantly living in a state of starvation from poor eating habits or anything of that nature, those people are going to be more likely to break down proteins. So in that sense, we'll start producing more urea cycle enzymes. The last part of this chapter looks at specifically how we break down amino acids. All of our amino acids are going to be broken down into alpha keto acids that can work as intermediates within other metabolic pathways. Some of our amino acids will produce more than one intermediate, and I've colored those in the slide here in red. We have 7 ketogenic amino acids. These are going to be amino acids that ultimately will form acetyl-CoA So they can form ketone bodies. That's why they're called keto. Remember, we form ketone bodies during times of when we have very little sugar. We're breaking down fats. Well, in that same situation, that's when we may begin breaking down amino acids as well. Our glucogenic amino acids are those shown here in this tan color. 6 of our glucogenic amino acids can form pyruvate. 5 can form alpha-ketoglutarate. 4 can form succinyl-CoA. 2 can form fumarate, and 2 can form oxaloacetate. This particular slide is a summary, and I think this is probably a good way for you to study. Those of you that don't like pictures, I also made you a list so that you could try to keep up with this. What I do not want you to do is try to memorize all of the degradation pathways. My hope is that you remember enough about the structure of your amino acids and you know enough about the structures of these molecules within the citric acid cycle that you can almost predict what we would be able to form based off of those structures that you know. What I would like to do, though, is go through some of these breakdown processes with you just so you can kind of get an idea for how they work. Again, you're not expected to memorize any of these. So this first one is showing you how we degrade some of our ketogenic amino acids. So we'll break down tryptophan, releasing alanine, ultimately pyruvate. We also produce some intermediates that form acetoacetyl-CoA and acetyl-CoA. Same thing with phenylalanine and tyrosine. We form fumarate. We form acetoacetate. So ultimately, these very large amino acids with your benzene rings, they're going to form acetyl-CoA. So these are going to be the ketogenic amino acids. This slide shows you amino acids that can break down to form alpha-ketoglutarate. We can break down arginine, histidine, glutamate, and glutamine to form alpha-ketoglutarate. We can break down asparagine to aspartate simply by losing an amine. Then aspartate can be broken down into oxaloacetate. We take leucine, isoleucine, and valine. We break those down. We do this a lot in the muscle to produce fuel for energy. Some of our intermediates from breakdown of tryptophan, not only do they form our ketone groups like acetyl-CoA and acetic acid, but sometimes we'll break down tryptophan into other molecules. Just to give you some examples, one of the pathways to produce niacin that's part of our production of NAD comes from breakdown of tryptophan, as well as a neurotransmitter, serotonin. There are multiple defects in the breakdown of amino acids that can lead to specific diseases that are unfortunately more common than we may like to believe. A very common one that a lot of people recognize is PKU. PKU stands for phenylketonuria. This is a defect in the ability to break down phenylalanine. A lot of times babies are tested for this. You'll notice on the back of some of your soft drinks, it'll warn you that if you suffer from PKU, you don't need to ingest this particular food. The only thing we can do for it is tell people to limit their phenylalanine intake. I think I may have another slide. Yes. So people that have PKU, they don't have the ability to do this first step in the breakdown of phenylalanine into its ultimate breakdown products.